A very good afternoon and hearty welcome to all. Myself, Dr. V. Bhuneshwari, working as Associate Professor and Head in Biotechnology UG, Kongunada Arts and Science College. As one of the alumni of this prestigious institution, and on behalf of Department of Biotechnology and Management of Kongunada Arts and Science College, I feel proud to welcome you all for this international alumni webinar series on insights into translational research and its application, which is to be conducted from 3/6/2020 to 8/6/2020. It's a pleasure to organize this alumni webinar series as per the suggestion and motivation from our respected Secretary and Director, Dr. C.A. Vasuki ma'am, who has given the platform to share the knowledge among us. And it's our proud moment to have our prestigious alumni research persons from different countries such as London, Singapore, Taiwan, Switzerland, Thailand, etc. Thank you, ma'am, for this opportunity. Now I request Dean Academics and Head PG Department of Biotechnology, Dr. S.R. Madan Shankar sir, to welcome the gathering. Sir, please.
Good afternoon to all the participants, and Shri Gurubhyo Namaha. At the outset, it's my pleasure and privilege to welcome the speaker of today's session, Dr. Kalpana Surendranath. She is currently the leader of Genome Engineering Lab, University of Westminster, for having consented to be one of the resource person in our webinar series on translational research and its application. I welcome you, Dr. Kalpana. It's my pleasure to welcome our Secretary and Director, Dr. C.A. Vasuki. She has given a lot of support in organizing all these programs at our campus. I welcome you, ma'am. I also welcome our principal, Dr. M. Lakshman Swami, and the HOD for most of us in this batch, because we all had our beginnings, learnings and biotechnology from him. I welcome you, sir. I also welcome Dr. Bhuneshwari, Head of Department of Biotechnology, and all the faculty members who are present here for this function. I also welcome all the participants who have registered for this webinar series. And with this, it's my pleasure to introduce my classmate, Dr. Kalpana Surendranath. She is currently the lecturer of Life Science and leader of Genome Engineering Lab, University of Westminster. She is an established researcher in the field of DNA repair and genome stability. She has introduced CRISPR genome engineering technology to both teaching and research in the University of Westminster since 2017. And ever since the introduction of this concept, her laboratory has won a lot of accolades in the subsequent years, where she has received about 125 funds, genetic summer grant fund, and to name a few. Dr. Kalpana also runs the Institute of Biomedical Sciences, accredited short course in CRISPR genome editing. She has received notable awards and grants throughout her research career. The first one, I would say, started from our campus in Kongunadu. She was university rank holder in her undergraduate program. Thereafter, in 2010, she received the Quintin Hawk Trust Award in 2020. In 2019, the Genetic Society Award, and so on and so forth. She conceived the Gene Editors of Future Student Training and Development Program and awarded with 25,000 pound in 2020. She has also received the highest honor of the university, the Vice Chancellor's Westminster Award, Woman of Westminster Award, and Individual Award for Teaching Excellence in the year 2018 to 2020. Having her today is our pleasure, and it's quite nostalgic because when we studied here in this campus, we used to literally fight for notes. We used to literally fight for little marks, say even half a mark or even 1 mark we used to fight, and that was a healthy atmosphere which was created in our campus. And being the first batch of students, we had lot of privilege in using the laboratories, because I would always say all our development started from here with this laboratory, and this laboratory was supposed to be the best in 96, when no other college even thought of establishing a beautiful laboratory of this kind. So all of us are privileged to be alumni of this great institution, and it's again a proud moment for me having come back to my alma mater as Dean Academics and PG Head of Biotechnology, where a request to most of the alumni who are here in this workshop is we will have to create a consortia and see that we contribute back to our alma mater in terms of academics, in terms of research, in terms of some collaborative programs in the future. Thank you all, and it's my pleasure to welcome all of you again for this program. Thank you.
Thank you, sir, for your warm welcome. Now I invite our beloved Secretary and Director, Dr. C.A. Vasiki ma'am, for the inaugural address. Ma'am, please.
Good afternoon to everybody. I'm sure good morning to Kalpana.
Morning, ma'am.
Must be a very bright, beautiful morning because it is summer.
It is. It is.
It is nice in a bright day to meet all your friends and your your friends from your college and your alumni friends. So it must be a very bright day, even a brighter day for you.
Indeed.
Yeah. I'm very pleased on behalf of Kongunad Arts and Science College to invite our dear alumni, Kalpana Surendranath, if I'm correct.
Yes.
Sorry if I pronounced it wrong. Who is the first batch of student in 96, if I'm correct. Even though I was not here, you were literally, I would say, mollycoddled by my father, who was then secretary, because he found the Department of Biotechnology was the most prestigious department, and he felt like, they were his own children. That's how he developed all of y'all, and I could see the outcome of it. Like all of y'all today, the first batch students have bloomed really well in all areas, and quite a few through Madan Shankar, through Dr. Madan Shankar, who is also a first batch alumni. I came to know that all of you all are well placed in different parts of the world. I'm sure, my father would be very happy if I mentioned this to him, because he always used to be very proud about the biotechnology laboratory, what he created. Even in those days, it was about more than nearly 1 crore in 96. That was a big money. And in fact, I was, when I came from UK, I said, do you think this much is necessary for today in India, this much research? And this is plainly, I'm telling the truth. I was not very happy. I said, you should develop other subjects which needs less investment. But one thing I learned from my father, he said, educational institutions are not investments to look for profits. They are knowledge imparting places where you should make sure one day, I will definitely, my students will bring Nobel Prize to these institutions. And I feel now, after coming and taking over as secretary of this college, I feel that's really what he said made sense to me, especially after seeing all my alumni who are placed in good position. And you are one, Kalpana. I think
you
get the Nobel Prize for this institution. I'd be very proud to say that. So today, I think with this small brief thought, nostalgia, because I am also an alumni of this institution. And so I also have great dreams for this place, Kongunad Arts and Science College. And most of the people who have come here, I would like to say it our college is one of the top 100 colleges in the country. And Also, like, we had achieved lot of awards from UGC, and one which we are very proud about is the College of Excellence, which has been given by UGC. Only 19 colleges have been given throughout India, and we are one among that. And when I tell you the number of colleges in India is more than 40,000 colleges. So it is mainly for the contribution of our teachers and our students, all the stakeholders for that matter, are really the ones who have contributed for this institution to grow to such great heights. And today's webinar, which has, I should actually thank the webinar, because today more than 3 webinars have taken, is going on in our college, and people are talking from all over the world. And this webinar has brought together people from far and wide places, because we always thought, inviting a person from abroad, so many processes has to be undergone, expenses to be borne. But now when you see, webinars have made it all easy. And now people are getting used to this technology. And from now on, I think they will all of us are going to use this very commonly, these webinar sessions. And through this webinar, today, our Department of Biotechnology has organized this series by our alumni, the insights into translational research and its applications, which is a very excellent topic they have taken. And when you see what is translational research, this is what, when we studied, that is in the 80s, 70s and 80s, a common term what we used as, at that time, we never even had a subject called biotechnology, for your information. But when we studied, we commonly used the name lab to land. All your research, whatever you do, it should reach the common man. So that's exactly what in translational research is in biomedical domain in recent years. That's what you call. It is usually an attempt we bridge between the gap between the knowledge that is produced in the laboratory to the clinical bedside. So most of your biomedical research and biotechnology research, biochemistry, all this are clinical based, and these should reach the common person, common man, that is in the clinical bedside. So that's exactly what today you call, in a very posh way, the translational research. And it is actually any research, when you see, it claims to help the society to harvest the benefits of its investments in the scientific research. See, we invest crores and crores and billions of pounds and dollars in research, whether that reaches the society. So that's one of the reasons today man is very confident that we can find solutions, and for all even for our COVID, which is now the entire world is being tortured, to be true. The scientists are ready. They're very confidently saying we can come out with an answer. It is all because the investments that was being done in the past. So in this web series, I'm sure the range of topics will be discussed to cover the major areas of science, from genomics to genome editing, cancers to stem cell therapy, neuroscience to novel drug discovery systems. So when you see in future, for these young students who are listening, I'm telling, the future of translational researches of all, how is it going to take? It should be very adaptable, and people should be ready for lifelong learning. And they have to be highly curious about a lot of different things, like collecting data and ideas from basic literature, and creatively applying these to present disease solutions. This means you have to think outside your box, thinking outside your comfort zone. Like translating is exactly what scientists do, like taking information from one domain and expressing it to another, and communicating daily with people who speak different scientific languages. Like, for example, the laboratory scientists should be able to have a dialogue with the clinicians, the patients, the biostatisticians, the epidemiologists, the pathologists, and the patients. So that's how the knowledge should be translated throughout the chain. Our translational scientists also should be able to move from always from basic research. So it doesn't mean when you become a biotechnologist, you should lack the understanding of basic research. So basic research to clinical application, and again, they should be able to come back to the lab to inform more basic science. So when you see all these different branches of biological sciences, and today, whatever the sciences that are contributing to the human mankind, all started and branched off from basic science. Let me tell about Albert Einstein, what he felt. Learn from yesterday, live for today, and hope for tomorrow. And this is exactly what I think our alumni have learned from our institution and are holding prestigious positions today. And I hope they will contribute to the better tomorrow and serve as beacons for the juniors as well. Newness and novelty has been an intriguing and fascinating since Neanderthal times. Now, it is excitement and exuberance, surprise and science, like our COVID, cheerfulness and joy, hope and happiness, boldness and buoyancy, positiveness and passion. So I join you all through this webinar series to explore the experience and the latest development in science and technology. So thank you, and we are looking forward for a wonderful session by our alumni. Thank you.
Thank you, ma'am.
Thank you, ma'am, for your motivating and kind words. Now I invite our dedicated principal, Dr. M. Lakshmanaswamy, sir, for felicitation. Sir, please.
Recommendation. Kalpana.
So, vanakkam, sir.
Vanakkam.
Sir has come in a
50.
I'm very emotional now. I can't explain in words. I'm like, I'm getting very emotional.
HOD. I think sir can remove your cap.
Yes, our guru, our kind teacher. I'm getting emotional. It's great meeting you, sir.
Thank you. Good morning. Good afternoon, all of you. Vanakkam, Kalpana, Dr. Kalpana, madam. நல்லா இருக்கீங்களா?
I'm நல்லா இருக்கேன் sir. I'm good.
நம்மள தான் ஒண்ணும் மறக்கலையேம்மா?
No. நல்லா பேசுவேன் sir.
Okay. எப்ப வருவீங்க இந்தியாவுக்கு?
We are planning for next year, so next summer. Next year வரலாம்னு plan இருக்கு.
Okay. Thank you. Most respected Patron Dr. C. Vasagi madam, Head Department of Biotechnology for PG, Dr. S.R. Madan Shankar, and first gold medalist, and Kalpana also the another gold medalist, and Dr. V. Bhuneshwari, Head Department of Biotechnology for UG.
She's a gold medalist as well.
She's also gold medalist. Now she's the coordinator for the iHub and also for the Institute Innovation Council. Very dedicated staff member of our college. Dr. Congratulations to Madan Shankar and Bhuneshwari, and the organizers, resource person, Dr. Kalpana Surendranath and Dr. Gandhi, a very sincere student in my class. He never speak at least even single word. That's a very innocent student. And Shibu also very sincere and dedicated student. And Dr. M. Giri Ramkumar and Dr. Prasant Mani, they are the best student. Once again, I congratulate on behalf of the management. Then coming to today's webinar topic, International Alumni Webinar Series on the insight into translational research and its applications. There are generally two categories of translational research. First one is basic to clinical. Already our secretary madam highlighted basic to clinic, and then second one is clinical to the population. Translational research seeks to produce more meaningful, applicable results that directly benefit human health. The goal of translational research is to translate basic science discoveries more quickly and efficiently into practice. Translational research is the process of applying knowledge from basic biology and clinical trials to techniques and tools that address critical medical needs. Unlike applied sciences, translational research is specifically designed to improve health outcomes. Look at drugs like the Gleevec, Avastin, and Herceptin. All are examples of the translational researchers converting molecular knowledge about specific cancer cells into effective targeted therapies. So with these few words, once again, I congratulate the biotechnology head of the departments, Dr. S.R. Madan Shankar and Dr. V. Bhuneshwari, and the most valuable resource person today. Thank you for the opportunity given to me, and I wish the program a grand success. Thank you.
Thank you, sir.
Thank you, sir, for your nice felicitation. Now I invite our prestigious guest of honor, Dr. Kalpana Surendranath ma'am, to present on the CRISPR revolution, rewrite the genetic blueprint of life, write a better future, ma'am. Ma'am, please.
Thank you. Thank you, Bona. Can you all hear me?
Yes, ma'am.
Yes.
Perfect. Perfect. So this is a very emotional moment, really. I totally went away from the topic and I feel as if I am at Kongunadu College. I begin the talk as a student of this college. I may work elsewhere, but I today talk as a student of this, a proud student of this college. I'm really emotional seeing Sir, Madan, ma'am, everyone start this occasion. So I went back to my student days. So it's an immense pleasure to be able to talk to you all today at this magnificent college. As a student, as I said, of this college, I would like to start by a few thank yous. First of all, thank you to the great visionary, our beloved secretary of our days, and Dr. Aruchami sir. Amazing the kind of support, just like respected Vasuki ma'am mentioned. I mean, I can't explain in words the kind of support that has been given. Biotechnology was still very, very new to even outside world at that time. When we did our biotechnology degree, it was like a dream course, and amazing. So thank you to the great visionary, again, respected Dr. Aruchami sir, and ma'am, respected Vasuki ma'am, an inspiring woman leader. I look up to you these days very closely, and I mean, the way you inspire, leave alone the alumni, every student, I can believe they are very, very lucky to be in the presence, just like Dr. Aruchami sir. So we are all lucky indeed. And thank you to our kind teacher, a patient, kind teacher, Dr. Lakshmanasamy sir. Thank you for tolerating all our mischiefs and still giving us and enlightening us with immense knowledge. And we learned a lot from you, sir. Thank you so much. And my competitor, my colleague, my friend, Dr. Madan Shankar. He has been a great inspiration to many students like me, how to be disciplined and how to be focused and how to live a great life, to be honest. So amazing, Madan Shankar. Thank you for getting me in touch again with our magnificent institute. So I look up to you and learn from you a lot. So thank you, Madan. And Bhuvana, thank you, Bhuvana. You've been so sweet in responding to my queries, and I mean, I have seen you as well quite some years back at the Indian Institute of Science, a very passionate scientist. The moment I saw Dr. Bhuvana, I felt that, I mean, she's going to shine in science. She used to work late night. She used to walk alone at 3 o'clock on the campus. A very, very dedicated scientist indeed. I mean, anyone would have spotted her shining in science. So there you go, Bona. You are in this magnificent institute. Good luck and all the best for all your endeavors as well. So that's a long note of thanks. But as I start, as I said, as a student of this magnificent institute, I believe that it was necessary to begin this way. And again, good morning, good afternoon, and good evening to everyone that is attending this seminar from different parts of the globe. I hope you find this useful and inspired, for the students in particular, inspired just as I got inspired 6 years ago. So I'm going to, there's some blank stuff that is running. So I'm going to stop my video if that is okay. And is that okay, Bhuvana?
Yes, sir. Yes, ma'am.
Yeah. So can you hear me now?
Yes, ma'am.
Okay. So I have a feeling that it's going a bit. Is it not breaking? Is it fine?
Yes, ma'am. It's fine. Ma'am, is it visible? The PPT is visible for you?
No, I can't see the PPT.
Ma'am, can you please pin the UA Biotechnology screen?
So, okay. Yes, I did.
Is it visible, ma'am?
No, I can see you and Matan.
Kalpana, now?
Not yet. It takes a bit time, Madan, if I'm right. It takes a little time to sync. So we may want to little bit wait. So as long as the participants see it, it's fine for me. I have a supporting.
It's fine. It's shared, Kalpana. Just check.
Not yet. Not yet.
You're presenting on it. Yeah, people can see. They are responding.
Okay. So I can't see.
Ask her
that's
to
fine.
Ask her to bring
That's
the
fine.
slide.
I have my slides with me. So
Okay, okay. Fine.
Don't worry. So I will prompt when I need it to be changed. Is that fine? All right. I did pin the screen. I can see Bona on the screen, but not on the.
We are not able to see you. Your video is on.
Your video is on, ma'am?
Let me check.
Listen now.
Can you see me now?
Yes.
Can you see me now?
Ma'am, now I'm sharing the PPT.
Okay.
Ma'am, could you see, ma'am?
PPT. Let me just pin. Yes, I can. Yes, I can. Perfect.
Okay.
See, put our video here.
Okay. Are we okay to start?
Yes, ma'am.
Okay, great. Okay. So I can see the slide, but I'm looking at my computer, so bear with me. Don't think that I'm looking elsewhere. I'm indeed looking at the slides. Okay. Ma'am gave a very good introduction about translation research. I was thinking of starting this seminar by introducing the difference between basic research and translation research. Ma'am used all the very nice words, lab to land, bench to bedside. That's what actually is translational research. So 6 years ago, I came across this powerful tool that has revolutionized several areas of biotechnology and biomedical sciences. And eventually, actually, what led to the creation of Genome Engineering Lab at the University of Westminster, where I work currently. So the tool is Crispr, and I'm going to share with you all some interesting facts about this and how I use this tool in my own laboratory. But put together This is a very powerful tool which allows researchers to easily alter DNA sequences and modify gene function. So that's it. So if the students that are attending this, if you think, is this the first time we ever started editing human genomes? The answer is no. We have been doing this since the 1990s, mostly by random homologous recombination. I'm sure you all would have seen overexpression of interested genes in various systems, but that was all a random process. But CRISPR is a very precise tool. To base pair precision, you can edit any genome of your interest. So that's the speciality about this particular tool. So can we go to the next slide?
Next slide goes on.
Can we go to the next slide?
Yes, ma'am.
Yeah. The previous one, please. The previous one. Yes, thank you. Overview. Yeah. So I understand that this is a gathering of student learners and experts. So I have divided this talk into two major parts. First, I will talk about the origins and fundamentals of CRISPR technology. And in the second part, I'll talk about how CRISPR is used in laboratories for biomarker discovery or validation of biomarkers. So next slide, please. Because of its sheer simplicity and reliability, the technology is one of the most widely used one in both basic and applied research laboratories. So undoubtedly one of the biggest science discoveries of the decade. So I'm very delighted that I get to work with it on a daily basis in my laboratory. So next slide, please. So what is CRISPR? Actually, CRISPR is an acronym for a very long name, clustered, regularly interspaced, short palindromic repeats. It's the way the sequence appears in the bacterial genome where it was acquired from. I'll tell you a little story about this in a minute. And Cas9, so there are two terms that are used in relation to CRISPR technology, CRISPR and Cas9. So Cas9 is the most commonly used enzyme. So Cas proteins are CRISPR-associated proteins. So it's a big mouthful. CRISPR is it stands for clustered, regularly interspaced, short palindromic repeats. So it's a big mouthful, so we call it CRISPR shortly. So next slide, please. Right. What is CRISPR? What can CRISPR do? So CRISPR is a molecular tool, basically. What is the nature of the molecular tool? Keep thinking. I'll come to that in a while. What can it do? This CRISPR molecular tool can target particular sections of DNA specifically and can make a cut, a precise cut at the target site. So what do we want to achieve by creating a precise cut on any genome? Two things: make a gene non-functional, mostly, or replace a new gene, or replace a dysfunctional gene with a more functional one. So these are the two things that we can do using the technology, to put it in a nutshell. Next slide, please. Right. I would like to begin the lecture with the applications of CRISPR. Why is CRISPR so greatly talked about? One may be thinking, what is so special about this technology? the speciality about this technology is the endless applications of it. So for greater appreciation of the limitless application of this topic, I have introduced this slide. So you can see, you can actually zoom and read at your own ease later. But I would like to very briefly mention that CRISPR can be used for treating serious genetic disorders. It can be used for increasing food supply or improving crops. It can be used to advance clinical diagnostics. Actually, today's talk is something related to clinical diagnostics, how I use the technology, the CRISPR technology, to validate a biomarker. So it can be used to advance clinical diagnostics. It can be also used for personalized disease treatment and creating various different human disorders. So also you can see there's a little elephant here. If you can see, it's a woolly mammoth. You can also bring back extinct animals to life. If you are the one that appreciates all these prehistoric creatures, CRISPR can actually make those things come back live. So it's a very powerful technology with endless applications. So I wanted to draw your attention to all these young budding scientists to think about which areas actually they want to explore to contribute to the betterment of humans. Okay, next slide, please. I particularly work, there are endless applications and, sorry, can you go back, please? Yes, thank you. So you can see that the technology can be used for many improvements that we want to make. But I use in my laboratory CRISPR technology for enhancing clinical diagnostics, and we are closely moving towards treating human beings and improving personalized disease treatment opportunities. So we are closely moving towards the clinics at this point. So the focus to put together is to create cellular models of human diseases. So, and I'm currently always actively looking for collaborations, and I'm open to discussion if anyone is willing to collaborate in this direction. Next slide, please. Okay, so let's begin by looking at the cells, the tiniest yet robust functional units that decide the fate of all multicellular organisms. So no matter, we are all in a stage of lockdown in terms of a social setup, but our cells never go into a lockdown until we disease. So, I mean, they are the most robust. I'm a big fan of cells ever since I became a cell biologist, cell and molecular biologist. I cannot stop admiring the way the cells are organized, and then the way they keep themselves busy, they talk to each other, really amazes me a lot. And let's begin by looking at the cells. So they are the ones that decide the fate of all multicellular organisms. And even more precisely, the fate relies in this tiny little piece, nearly a 2-meter length genetic material that is packed inside the nucleus of all eukaryotic cells. So the genetic material serves as an instruction book of life. And this instruction book is made of 3.9 billion letters. And you know ATGC, the combinations that exist in these letters. So this instruction book, as I said, is about 2 meters long, and it exists as a heavily compacted structure. That's what you see. So what you see in the slide from your left is a group of HeLa cells, and you see the nucleus stained with DAPI in the cells, and inside the nucleus is the chromosome, a heavily compacted structure. As I said, a 2 meter long DNA is compacted and packed in supercoiled and presented in the very tiny micrometer sized nucleus. Can you believe it? Amazing. So, and the chromosomes are made of nothing but proteins and the DNA. So this is what we want to, this is what actually we want to look at more closely in the context of CRISPR. So the genetic material is what actually acts as a blueprint of what it takes to build an organism. So all the instructions of our daily activities are inscribed in this instruction book. Any change that we create in this instruction book can be deleterious or sometimes beneficial. Can you please go to the next slide? Okay, let me give you an example of genetic change that can be harmful. So one may be thinking, okay, why we should think about rewriting the human genome? What's so great about it? Why should we rewrite the human genome at all? So I would like to come back to that point again, that genetic changes. So all the time, scientists have been looking for creating changes at the genome level to introduce new properties or remove harmful properties. So this is an example of a harmful change, a single typo. I can't see the slide. Can everyone see the slide?
Yes, ma'am, we can.
Okay, that's fine. As long as everyone can see it, it's okay. For me, it's a bit, I think it's a bit of a connection thing. So it's okay. I can carry on, right?
Yes, ma'am.
Okay, thank you, Puna. A single typo, A to T, is what leads to the disease sickle cell anemia, where the mutation A to T causes the changes in the conformation of the oxygen-carrying protein and distorts the structure of the red blood cells. The red blood cells become crescent-shaped as opposed to the biconcave disc shape, and now cannot pass through the vasculature as easily as the biconcave discs. Therefore, they are prone to damage. So patients that carry these sickle cells have their blood cells dying within 10 days, 10 to 15 days, as opposed to a 120-day lifespan of red blood cells. So next slide, please. Are the genetic changes always harmful? The answer is no. Genetic changes can be beneficial as well. Here is an example of a mutation in the T cell surface receptor, CCR5. So mutations in this receptor leaves the receptor in a shortened form. And in this short form of the receptor restricts the entry of a HIV virus inside the human cells, the T cells, therefore rendering protection against HIV infection. So that's one example of beneficial genetic difference. So in terms of plants, very briefly, mutations has been the key for plant breeding throughout human existence. You can see this is an example that is shown in the slide. So mutations that we introduced in a simple mustard plant has led to the development of various different varieties of vegetables that we consume. So mutations can be beneficial as well. Next slide, please. Has it changed? I can't see anything, so I'm just hoping that it is changing. So can you confirm that it is changed? Thank you.
Yes, ma'am, it changed.
Thank you, thank you. So, so where is CRISPR? So we know we want to create mutations, we want to create changes, we know it can be beneficial, it can remove harmful changes, but where CRISPR came from? So you may be wondering, where did we get this from? Did we design it, or did we find it from somewhere? So it's not an artificially synthesized tool. The answer is CRISPR is only an engineered tool. It existed indeed in one of the smallest living ancestors, the bacterial cells themselves. So just like us, currently in the state of this lockdown because of the virus, bacterial cells also are under constant threat of viral infections. So wherever there are bacterial cells, there are viruses. And so bacteria are under constant evolutionary pressure to evolve different methods to combat viral infections. So that's what is shown in this picture. So that's a single bacterial cell. So bacterial cells have evolved different methods. So they can either inhibit attachment, the viral DNA entry, or in the assembly. So bacteria have evolved many mechanisms to prevent viral infections. So next slide, please. And could you say just quickly yes once it's changed? Thank you.
Yes, ma'am.
Thank you. So, CRISPR is yet another system that is evolved by the bacterial cells to render protection against viruses. So please pay attention to this little circle. So that indicates a single bacterial cell. So, so the bacterial cell, upon invasion by the viruses, infection by the viruses, acquire a small piece of the viral DNA, and now inserts it into in their own genome in the name of a spacer. So that's what you see as a green box here. So a single section of the viral DNA inserted into a special locus called the CRISPR locus. So there are these black diamonds spaced regularly. So that's actually is the reason for this name. They are clustered. The black diamonds are clustered and regularly interspaced short palindromic repeats. So they are palindromic in nature. So the spacer is inserted in this CRISPR locus, and upon subsequent viral infection, now the spacer is transcribed into CRISPR RNA. CRISPR RNA, because of its high affinity to a particular enzyme called Cas endonuclease, can bind to it and now can degrade very specifically the subsequent invading viral DNA. So that's a very clever tool. So the bacterial cells have evolved this, and it's very striking that CRISPR is not, again, I would like to mention that CRISPR is not just a 1 day or a 1 year's discovery. It's a discovery that has followed, I think, over the couple of decades from now. The first report of CRISPR repeats occurred, as I mentioned in the slide, in 1987. So you can imagine how old. So people have been able to gather this knowledge. The beauty is that people have been able to gather this knowledge and design a tool that can be used to edit genomes in general. So about 40% of the sequenced bacterial genomes and 90% of those from RK contain at least one CRISPR locus. So you can imagine how abundant this is represented in the microbes. Okay, so next slide, please. Right.
It changed, ma'am.
Thank you. So CRISPR is, as I said, is a result of decades of work by basic researchers all around the world. And these are the two, leave me, that's Kalpana there, just standing next to the inventor of this technology, Professor Jennifer Doudna. Got to meet her in the Royal Society meeting recently. And two women, so scientists, Professor Jennifer Doudna on top, and Professor Emanuela Charpentier at the bottom, contributed immensely in the biochemical characterization of the Cas proteins. And she believes indeed that the future of medicine may land within 5 to 10 years, but it's not an easy job. It's going to be a tough task indeed. So many of us feel excited about the opportunities. There's a lot of motivation, both on the part of academic scientists and also companies. But we need more and more research first to understand this tool better, to be able to find new applications for sustainable use of this technology. Next slide, please.
Change, ma'am.
Okay, so that's Feng Zhang. There are two contributions. There's a reason why I put these pictures. One, on one side, we all think sometimes very less, oh, microbiology, oh, little enzymatic test in the lab. But these are very important. Every small experiment we do in the lab, be it good laboratory practice, just arranging, keeping your workbench in order itself is a skill, is a graduate attribute. So, I mean, we all overlook simple things. We think because we get things easily, it's so simple. Why do I focus on it? But these guys are the ones actually that focused on such very simple finding and came up with, they are the biggest, I think it's rushing for a Nobel Prize very soon, CRISPR. So we will see these names on a Nobel list very soon. So you can imagine they used very simple biochemical techniques, biotechnology methods. So this guy is a biotechnologist. So he actually has generated methods for engineering human cells. All the protocols that I use in my laboratory are designed by Feng Sang. So you can see the reference. There's a very useful reference I put on the slide. I follow most of the protocols created by Feng Sang. Next slide, please.
Change it, ma'am.
Thank you. So there are two major components. So what makes a CRISPR tool? There are two major components. We are moving slowly from the basics of the CRISPR tool into the mechanism of how the CRISPR tool functions. Very slowly, we are moving into that, just to let you know. What makes a CRISPR tool? It's a RNA protein complex, very simply. An RNA protein complex, a very big protein, about 160 kilodaltons, 160 kilodaltons, Cas9. It's a huge protein. You can see a big bulk structure here, colored in red, and a very small guide, just 20 base pair guide RNA. That's it, really. RNA, a protein RNA tool. Next slide, please.
Change it, ma'am.
Okay, so so we always talk about CRISPR in the context of Cas9, and Cas9 is not the only protein. There are also other Cas proteins, but these Cas proteins share a similar. So don't be overwhelmed by looking at this thing. What I want you to understand is just the two domains that are common in these Cas proteins are the recognition lobe and the nuclease lobe. There are the protein, actually the Cas proteins are organized in 3 dimensions into two lobes, the recognition lobe and the nucleus lobe, and in between which is sandwiched the DNA. So that's what I want you to appreciate in this slide. And also how different Cas proteins can be in terms of the recognition. If you look at, simply by looking at, you don't have to look at the ATGCs and the details, but just by looking at where it's the little arrows here indicates the cutting site. You can see the cutting site differs, the way the recognition occurs also differs across Cas9 proteins. So Cas proteins, sorry. So though throughout the talk I'll be referring to Cas9, please bear in mind that there are also other Cas proteins which can be used in this technology. Next slide, please.
Change, ma'am.
Okay, thank you. So this is again to show how fast this technology is being used, how robustly, how widely this technology is being used in laboratories worldwide. Due to the potential of this technology, like simplicity, very reliable, being a very reliable tool, the number of publications have skyrocketed ever since 2015. So if you just follow from 2015, you will see that the number of publications have increased. And this is just up to 2018. But you can see it has already crossed over 4,000 publications in a year, and now it'll be even more. So the people are actually validating it, testing it for various purposes at the moment. Next slide, please.
Change it, ma'am.
Thank you. Okay, so we know what the technology is. It's a very simple molecular tool, RNA protein complex. What can this RNA protein complex do when we introduce it into the human cells? Next slide, please. Okay.
Change it, ma'am.
Got it. Yeah. So what can it do? So the green structure on top of this slide indicates a single human cell with a red colored nucleus. Just for understanding purposes, it's colored red. So when we introduce this CRISPR tool, there's a huge roadmap. So I'm not getting into that. Let's say if you introduce a plasmid, the plasmid has to be exported imported into the nucleus and then transcribed. RNAs are exported into the cytosol where it is translated, and then the CRISPR tool is formed. Again, the CRISPR tool shuttles back into the nucleus, and then that's where the recognition and the cutting happens. So it's a huge roadmap, literally, from the cell surface. The tools that you introduce, it depends on what tool you're introducing. There are many ways of introducing, so if you're interested, please look at it. I'm not getting into the details of it. You can introduce this tool as a plasmid vector or RNA protein complex or messenger RNAs itself. So there are many ways. So depending on the entry point, the roadmap differs. So what eventually happens inside the nucleus of this human cell is this. So you see the ash bars, that's the DNA, and the blue big bulky structure is the Cas9 protein carrying the guide RNA, which is in green. So the scissors indicate the location where the Cas9 cuts. So this is all the CRISPR can do. All it can do is to scan, once inside the nucleus, the CRISPR, the Cas9 RNA complex can scan the genome, find the specific site, bind, and create a double-strand break. Now, very specifically, 3 bases away from a motif called PAM motif. So very, very specific. I didn't believe it until I tested it in my laboratory. I was like, okay, people claim a lot of things. It's not going to happen, because I was just setting up my laboratory 2 years back. And I was thinking, okay, let me see if it works. Indeed, it occurs. In this 3.9 billion base of the human genome, we created a specific cut in 2017. It was a big moment for me, though it's very simple at this point. But at that point, it was amazing. It creates a specific double-strand break 3 bases away from the PAM motif. Next slide, please.
Change it, ma'am.
Thank you. So yes, okay, we make a double-strand break. You may be wondering, what happens after this? What happens after this? We don't have a control on what happens after this, because the cells take over, because the cells are under constant threat, both by internal as well as external sources of DNA damage. So just to let you know, an interesting point here, a single human cell incurs about 1 million DNA damage every day. Can you believe it? 1 million DNA damage. So it can be single-strand breaks, double-strand breaks, base modifications, and so on. So double-strand break of all the DNA damages is the most toxic of all. So you can see it is reported that a single double-strand break, if left unrepaired, can kill a cell. So it's the most toxic of all DNA damages. So how does the cell now, okay, you're introducing an enzyme inside the human nucleus and creating double-strand breaks frequently. So the cell has multiple pathways to fix breaks. So the pathways that work, so please focus on the little boxed area. The pathways that work to fix double-strand break are 2. So either it is fixed by HR or by NHEJ, homologous recombination, or non-homologous end joining. So you can look at these little players at your own ease. But what is the take-home message from this is that the double-strand breaks are repaired by 2 pathways. One makes errors, other one is error-proof. So what happens when we introduce an enzyme that continuously cuts the DNA? The cells are in a rush to fix it before progressing to the next stages of cell cycle. And in that rush, they bring in more pathways because HR is the preferred one, HR is the error-free one, which can get back the DNA in its original form. But that always doesn't happen because by sheer overexpression of this enzyme in the human nucleus, we force the human cell to take a decision to bring in more pathways to fix the damage, because as I said, it can kill it. So it's sort of a SOS situation that we create inside the human cells. So NHEJ comes in. So HR, as I said, it's an error-free process, which can repair the DNA back to exactly how it was. But NHEJ is sort of works like a tailor. It can do patchwork. It can just trim the ends and attach it. So you can imagine the kind of changes NHEJ can introduce into the target site. So it can introduce mutations, it can introduce insertions, deletions, therefore leaving the gene disrupted. So this is what can happen. Next slide, please.
Go ahead, ma'am.
Yes, thank you. So using this technology, we can introduce as many as 13 different modifications in a single coding gene. For example, here shown is the single gene on the exon and intron organization at the genome level. So you can see, as indicated, you can cause mutations, insertions, and deletions. In the boxed area is what you see. We target exon 1. So exon 1 is boxed in this because exon 1, the gene can be coded by any number of exons. But when you want to create a knockout, early exons are the targets because we don't want any functional region of the protein being expressed. So CRISPR can be used to create 13 different modifications in a single gene. So that's the message from this slide. Next slide, please.
Change it, ma'am.
Thank you. So how is CRISPR? So now we are moving to the actual theme of this series, translational research, and how is CRISPR applied? I would like to move from this part from now on till the end of the talk, a little workflow. I want to provide you a workflow of how CRISPR is applied from bench to get a technology, to get an idea from bench to bedside. So the beauty of CRISPR is that, like respected Vasuki ma'am mentioned in the beginning, there are two branches of research, basic and translational research. So basic researchers actually don't end up in meaningful or it's not mostly or directly translated into clinical applications. Whereas translational research on the other side leads to the development of new diagnostic tests or drugs and so on. So CRISPR comfortably sits in between both. You can see that's what I want you to focus on this, dear students. So you can see CRISPR sits in both accelerating in the direction of accelerating basic research as well as in supporting translational research. So it's really a very, very, very useful tool. Next slide, please.
Change it, ma'am.
Thank you. So, so what do we want to do? So as I said, there are 13 different modifications. It's going to be a bit confusing. I want to tell you two major modifications that are actually done using CRISPR in laboratories worldwide, either for either for gene silencing or for introducing new copies of genes. So you can see the pathways that are preferred for gene silencing is NHEJ, the non-homologous end joining. So in these little boxes, you see either CRISPR can be either used for gene silencing and or introducing new genes. So two major applications. So for gene silencing, the pathway that is preferred is NHEJ, the non-homologous end joining, because we targetedly want to disrupt a section of the exon. On the other hand, the pathway that is preferred is HR, homologous homologous recombination. So, or also called as HDR. So, so all these applications of broader applications of CRISPR relies on majorly on these two methods. Okay, next slide, please.
Go ahead, ma'am.
What is the workflow? So you may be wondering, okay, now I know what CRISPR is, I know what can it do inside human cells. So how do I get it working in my laboratory? There are 3 stages of, that's how I see it. So there are 3 stages of a CRISPR workflow in the laboratory. The first stage involves designing the guides, so analyzing the target genomic location, designing guides, synthesizing the guide oligos from a service provider. Guide oligo is a 20 base pair segment, a very small fragment of nucleotide. So it's very, very small. So so we get it like the primers that we use for PCRs and stuff. We can get it synthesized from a service provider, and then we clone it into a vector. So I use very extensively a vector called PX459. It's an all-in-one vector. You can express this vector in bacterial cells, human cells. So it contains all the selection markers and all the elements that is needed for transcription and translation. So you can see, so it's a huge vector. So because of all these elements embedded in this all-in-one vector, it's very huge. It's a 9.4 kb vector. So we clone this tiny piece of the guide, which is about 20 bases, into this 9.5, one of the trickiest clonings you will come across. But I have standardized quite a few excellent ways of doing it. We can do it in no time in my laboratory. After we clone this, we sequence the plasmids, and then the plasmids are ready for now transfecting into human cells. So the stage 1 takes about, I can finish it in 1 week if I work, if I myself get onto the table and work. So if we have to redo certain steps, we manage to do it in 2 weeks' time. But that's very simple. Stage 1 is quite simple. Stage 2, because stage 1 is simple because it's all in the bacterial cells, so it's simple. So when it moves to human cells, it takes time. So you can see from transfection to clonal isolation is what we identify as stage 2. And it takes about 3 months. So we transfect, we throw the plasmids into the human cells, select them with a selection marker. Puromycin is widely used. It's a very, very reliable antibiotic indeed. And then we clonally isolate the cells. I'll show a little illustration that will be more clear to you. Stage 3 can extend for as long as the molecule is interesting to you. So you will know, as students of biotechnology, you will know we still work on P53. P53 is known for decades. So depending on how interesting the molecule is, stage 3 can extend. So next slide, please.
Change it, ma'am.
Thank you. So the first genomic target, so how did I use CRISPR tool, and what did I do with the CRISPR tool in my laboratory is to work on a breast cancer driver gene. So this finding came out of a clinical study. I have provided the references at the top of the slide. So I think the cohort of patients, about 2,000 patients, it's a large cohort, so it's a very significant piece of study. So we were quite convinced to work on this molecule. So, so this gene, the location of this gene is Q24.1 on chromosome 14. That was our first genomic target. Chromosome 14, if you just some fun facts here and there, because we are all sitting in our own isolated places. So just a little interesting stuff. Chromosome 14 is about 105 million base pairs. Mind it, dears, 105 million base pairs. So it's a very, very, even bigger ones are there, but it's a long one. It's a relatively bigger one. And we created in our laboratory without, because when I started my laboratory, I just worked with, I had 2 PhD students and 7 undergrads and 4 master students. So none of them had any experience in using this technology. I'm bringing these facts inside, hoping to instill some interest and inspire you all to use every little opportunity that is provided at Kongunadu Arts and Science College. So I did this when we had nothing, to be honest. It may be a laboratory in London, but when I started, I had only a PCR machine and few basic things, a cell culture hood and few basic things. That's it. And the reason why I have highlighted this in yellow is that with no big expertise and really taking students actively on board, training them, we managed to create a very significant 100 base pair change, in a human chromosome, which is 105 million base pairs. That was a big achievement. I still believe, I mean, the research has gone, I mean, we have progressed much faster and we are big at the university at this point, but still I believe and I still relish this moment of seeing that we have actually, with the help of our students, did this. So it's a big achievement indeed. Next slide, please.
It changed, ma'am.
Thank you. So just to visually show you, so I showed you a quick overview of what this technology takes in terms of different stages, but I want to show you all a little more in detail in the next few slides. Stage 1. So you can see it involves the guide designs. This is a guide. So the little box here, the little rectangle shape, shows 3 different guides that we designed for our first target. And so the exon architecture was shown here as well. So there are 2 exons for this gene, exon 1 and exon 2. Exon 1 is very, very small. So it's about 50 bases. So we didn't target exon 1 because there was no functional domain that is coded by that exon. Exon 2, we targeted the early part of exon 2. And next slide, please.
Change, ma'am.
Thank you. So from the top, so can you go back, please?
Yeah, it's changed, ma'am.
Thank you. Sorry, I wanted to just, so I missed out one point. So from the top, you see the exon A, panel A is the chromosome, and the boxed region is the location of the gene. And below that is the exon architecture. And below that exon architecture is where I have indicated the guides that we see synthesized. And we have provided an example of an illustration of how the guide is expected to bind at the target site. Thank you. Next slide, please.
Yeah, it changed, ma'am.
Thank you. So the little 20 base pair guides are then cloned into a plasmid. What you see is actually the sequence of the plasmid, PX459. So from the bottom, let's go from the bottom. So you will see the site of clone, the organization of different sections of the plasmid and the cloning site. So we use BBS1 and we clone the guide inside the plasmid vector. Next slide, please.
It's changed, ma'am.
Yeah, thank you. So now, so what do we do after cloning? It's easy. I'm sure you all will be doing colony PCRs. We do a simple colony PCR to look for the positive clones containing the guide of interest. And so you can see these little bands in the agarose gel, the top one, indicate the amplification of the guide specific sequence in a colony PCR. And we choose these colonies. So there are different, all these are like individual colony PCRs. There's so many that I've represented here. It's about a 250 base pair region that we amplify. And the positive colonies, you have to be a bit careful because the CRISPR plasmids are quite labile. They go through a lot of recombination. So you can't be 100% confident that if you see a positive colony, you will have a plasmid inside it. So we sometimes, for unknown reasons, we also find unusual changes in the plasmid. So we choose usually quite a few colonies, and then we clones, and then we sequence them. We send them for Sanger sequencing and verify them. So next stage, next one, please.
It's changed, ma'am.
Yes, yes. So the next stage is stage 2, is what actually is shown here. So once we confirmed that we have cloned the guides into the plasmid, we introduce these plasmids. So from the top, if you can follow me from the top, so those are 6-well plates where we grow human cells, and we introduce the plasmids into these human cells by different methods. QIAGEN's Effectine works quite well in our hands at this point. So we introduce the plasmids into the cells plated on 6-well plates, and followed by a selection process. We select these cells using puromycin, and after that, I'm sure you will understand what selection means. So the cells that contain the plasmid can grow in the presence of puromycin, whereas the cells that do not have the plasmid will die. So that's the basic selection process. I'm sure you all know that. Puromycin acetyltransferase is the resistance marker in PX459. So after selection, what we do is we grow the cells to congruency in the presence of the selection marker, and then we dilute the cells into monoclones. So after selection, the total pool is the parent pool. We call it as a parent pool, so total edited population. But a total edited population can be a mixture of hundreds of different edits. So that will not be useful for a researcher. We need to isolate the clones into single edits, which will lead to meaningful analysis later on. So that's what we do by more limiting dilution. We isolate the clones to single cell in a well, grow them, amplify them, and then we have a monoclonal population with one particular edit. Next slide, please. So that's what happens in the stages below. In the little 96 well, 24 wells, 6 wells. So you'll see different wells. So each of this well indicates, so from the 96 well, sorry, I started talking on the same slide, bear with me. So I realized that I need to quickly mention this. Single cells separated on a 96 well plate and then expanded in a 24 well and then onto on a 6 well, and then we are ready for the analysis. Next slide, please. Thank you.
Yes, changed, ma'am.
Thank you. So that's how the clones, I'm going to show you in the next 2 slides, how the clones appear after selection. So this one shows you how the clones appear after a day, 7 days, and 12 days after selection. You can see very few cells in the first day, and then they grow a little bit more in 7 days, and then in 12 days, quite to a quite larger patch. So next slide, please.
Yeah, it's changed, ma'am.
A bit. Thank you. Sorry for bothering you this way. I can't see anything. Sorry. It feels a bit every time. Yeah. So so you can see that this slide again shows you the different stages in a fluorescence system. This will be a bit more appealing to you. That's why I put in 2 different versions, a bright field version and a fluorescent version. You can see that the cells are in 1 day are few in number, as opposed to 7 days and 12 days. You can see a colony that is bigger. By 7 days, usually, if the colony is not very damaged, you can quite see around 100 to 200 cells in a colony, and which can be even more amplified after a week. So next slide, please.
Yeah, it's changed, ma'am.
Yeah. So what do we do? Okay, we have diluted the clones, we have grown the clones, and now what do? We analyze the sequence. We analyze the genomic sequence of the clones. So that's what I have shown you here in the slide. On top is the cells that are grown on a dish. Usually, 10 centimeter dish is good because you'll get good amount of DNA. And you extract genomic DNA. So that's what I have shown in the gel, which is right below the Petri dish. It's a cell culture dish. I wouldn't call it as a Petri dish. It's a cell culture dish. So you will see that the genomic DNA runs quite high in this agarose gel. So you will see I have isolated genomic DNA from different clones, and all the clones have provided us quite a good amount of genomic DNA. And having the genomic DNA alone doesn't tell us anything, because as I said, it's a huge stretch. It's a mega stretch of genetic material. What we want to do is look at that particular section that we edited. So what we do is a genomic PCR, and it's very simple. Again, you can see, I'm sure you will be aware of that. So we amplify a less than 500 base pair region of the target site. Sometimes, if you're lucky, if the changes that you introduced are larger at the genome site, so you will see that, for example, truncations of 100 base pair can already be visible. So that's what you see in this white little box with the agarose gel inside. So you see the wild type running higher, whereas the truncated clones have a shortened version of the truncated site, already giving an indication, before even we set out sequencing them, already giving an indication that we have a truncation there. So it was indeed, we were lucky to find this truncation at the first stages. So next slide, please.
Yeah, go ahead, ma'am.
Yeah. So we verified this. Okay, we do see changes in the agarose gel, but that's not the end of the day. We need to confirm it. We use both NGS, next generation sequencing methods, and Sanger sequencing. First, we attempt is to do Sanger sequencing, because Sanger sequencing is quite quicker and cost effective. So we go for Sanger sequencing to begin with. And so luckily, in our case, in the first rounds, we actually got good Sanger sequencing results. But some clones can be challenging, so we will be forced to do either AmpliSec analysis, which is the NGS based method, and can be a bit more expensive and time consuming. But as you can see, I have shown you by arrows the region that is deleted. You see that there's a gap in both the Sanger sequencing results that I have shown here. After the arrow, there is a gap which indicates the deletion that we have made. So this is what I proudly mentioned, starting from nothing. Our students have created 100 base pair deletion at this at the 105 million base pair chromosome 14. So but this is not the end of the day. So Sanger sequencing is just one method. One can also use NGS methods, which will give you more insights into the variations there are in the human genome. Next, please.
Yes, ma'am.
So Sanger sequencing confirmed that. So again, just to remind you all, I'm telling you the story of the first ever target that I deleted using CRISPR technology, CRISPR technology in my laboratory. So I'm just taking you through it step by step. So at the moment, I'm just trying to show you, yes, I confirmed the deletion at the target site using Sanger sequencing and simple agarose gel. But did I see this translate in terms of protein? The answer is yes. So you see that in this is a result of a Western blot, everyone. So it's not, don't be mistaken that it is an agarose gel. It's a Western blot. So you see, you can see the top band is a protein that is closely related to our protein of interest. So, but you can use that. That was not our target. So you can use that as a loading control. The one that is mentioned as ZFP36L1 is actually the one that we truncated. So please follow it from the left, so from the right of the slide, from the right of the slide. So you will see the full-length protein expressed wherever we haven't deleted it, whereas in the clone that has been knocked out of L1, there is no L1 protein. So you can see clearly there's no protein. And the truncated one runs shorter than the full-length form. So as seen by the smaller bands, I think it's about a few kilodaltons lesser compared to the full-length version. So because you can imagine 100 bases, about 33 amino acids, let's take on an average, 33 meaning about 3 to 4 daltons, yeah, a few kilodaltons missing from there. So yes, we confirm that we have created a knockout. Next slide, please.
Go ahead, ma'am.
Yeah, so CRISPR is just a tool. It can just kickstart a study, provide you a model which you can investigate. So the methods that we use for studying the model varies. So that's what actually is stage 3, is the functional characterization. So in our case, we characterize this molecule purely based on the nature of this protein and also the hypothesis that we made about this protein. So just to briefly mention you again that we are investigating a biomarker, a disease biomarker, in this case a breast cancer disease biomarker, and you may be aware that biomarkers play major roles in medicinal biology. They help us in early diagnosis, disease prevention, drug target identification, drug responses, studying drug responses, and so on. So that's what I've shown here. Biomarkers can assist us to develop diagnostics. Biomarkers can also help us predict the effect of or the response to drugs. So, and also biomarkers help us identify synthetic lethal interactions. So if whether or not the given molecule works alongside another molecule in a pathway. Yeah. So can you just a minute? Just give me a minute. There's a bit of an interruption. Yeah, there's a work next door and I can hear constant noise. It's a bit disrupting. Okay, back in the presentation. So biomarkers can allow us to accelerate diagnostics, predict the response to drugs, and then look at synthetic lethal interactions and arriving at new drug targets and so on. So next slide, please. Has it changed?
Yes, ma'am.
Thank you.
Yes, ma'am. Go ahead.
Thank
Yes, ma'am. Yes,
Thank you. So, as I briefly mentioned, so the biomarker that we worked in the lab, the first biomarker that we worked in the lab is ZFP36L1. It's an RNA binding protein. I don't want to get into the full details of that. Just to mention briefly, it's an RNA binding protein, and it plays a role in post-transcriptional regulation of gene expression. So it binds to mRNA molecules, targets them for degradation, and so on. This has been known in the scientific community for a long time. But what we have done in our laboratory is, by a couple of analyses, we found strikingly that this protein has more roles to play. It is multifaceted as opposed to what it was thought before. we predicted a role of this protein in resolving or in preventing replication stress. So if you want to know more about how did we find it, I'm happy to talk later, but it's a big story again. So the main point here is, the biomarker that we investigate is an RNA binding protein, a well-characterized RNA binding protein, but we discovered a new role that is preventing replication stress in cancer cells. So, so just briefly in this slide, I have indicated the importance of resolving replication stress and associated DNA damages throughout. So accurate duplication of DNA and transmission of identical genetic information into the offspring cells lies at the heart of cell division. So I'm sure you all will recognize these different stages. G1S, G2M, the G are the gap phases, S is the synthesis phase, and M is the mitotic phase. So these lines here indicate different checkpoints that are in place to prevent the cells from progressing to the further stages of cell cycle, to prevent the cells accumulate DNA damage. This checkpoint signal the cells that you are carrying too many damages. You should be repairing it before you proceed to the next stage of cell cycle. Literally, they signal the checkpoint, the signal continuously. And if the cell fails to repair the damages, then the cell is not allowed to progress to the further stages of cell cycle. So that is also important. So I put these three scientists here who received, it's important for their important contribution and DNA damage response. These three scientists, Tom Lindell, Paul Modrich, and Aziz Sankar, received the Nobel Prize a few years earlier. I had the opportunity because for my postdoc, I worked with a big scientist in the field of DNA damage response, and I had the opportunity to collaborate with the middle scientist, Paul Modrich. So it's due to my maternity-related work. I had to withdraw from that work. I appeared. You can see in the PNAs edited by this Nobel laureate, Paul Modrich. I am with them in the paper. So it's amazing. So it's a great experience to be able to learn from them. So what did we do with these clones? Going back to that, how did we check the role of this protein in DNA replication stress? We first looked at the proliferation capabilities of these clones. So in this graph, what you see is the growth of wild-type clones as opposed to the truncated ones. So what you see here is the wild-type clones grow much quicker as opposed to the truncated clones. So definitely giving a clear indication that the cells are struggling to proliferate. So I'm sure you will agree with me that each day millions of cells in our bodies undergo division to support the growth and to replace lost or damaged cells in our tissues. So we need continuous cell division. If there is a hindrance to cell division, it means there is a problem. We are still investigating this. What actually is making these cells grow slowly? What actually compromises the proliferating capabilities of these cells that lack the full-length protein is what is the actual basis of further investigation. Next slide, please.
Yes, ma'am, it changed.
Thank you. So we found that the cells grow slowly. So we started looking, because we suspected and predicted, also had initial siRNA-mediated experiments and provided us clues that this protein could have a role in resolving or preventing replication stress. We looked at replication stress markers. So in this slide, I'm showing you one such marker that we tested. So the left side, the blue stained nuclei, or the nuclei of the human cells, the clones that we generated, So, so you can see that this is the truncated clone untreated, or that which is the DMSO, and treated either with DMSO or with 0.4 micromolar afidikoline. So what is clearly seen is this: you see these nice little punctate spots that is getting abundant upon treatment with afidikoline. That is nothing but a marker of replication stress, which is 53BP1, so highlighted in red. So 53BP1 is a p53 binding protein and is a crucial component of DNA double-strand break repair. So accumulation of 53BP1 indicates accumulation of damage. So clearly, this data indicated us, and also you can see in the quantification. If you are interested, please have a look at the quantification. There's a significant increase as opposed to wild type. In the truncated clones, there's a significant increase in the 53BP1 foci. As I said, genomic DNA is a very busy structure. Hundreds of proteins enter and exit the DNA in a millisecond basis. And 53BP1 is one of those proteins that is recruited to the sites of double-strand break, DNA double-strand break. So accumulation of 53BP1 is an indication of an accumulating double-strand break. So that's indeed a convincing finding that yes, in the absence of the biomarker that we edited causes increase in double-strand breaks. Next slide, please. Are we ready?
No, ma'am, just wait.
Okay.
Yes, ma'am, you can.
Thank you. So the next biomarker that we test are micronuclei. So micronuclei is a golden standard actually for genomic instability. It's a gold standard biomarker for genomic instability. And again, if you are wondering what micronucleus is, micronucleus is the name given to the small nucleus-like structures that form whenever the chromosome is labile, or whenever there is a break in the chromosome. So this little fragment of the chromosome is not incorporated because of it's floating freely. It's not incorporated into the daughter cells. Therefore, there's a new nuclear envelope kind of structure that is included in the around the chromosome, and exists as a tiny nucleus-like structure. So the appearance of nucleus is usually a sign of genotoxic event and chromosomal instability. So more micronucleus, more micronuclei. So there's a plural, singular micronucleus. So more micronuclei, increased genomic instability is the take-home message. So yes, we did see in our clones there is increased. So you see these tiny little budding structures. This is only, bear in mind, this is just a representative image. So we count more than 900 such nuclei to be able to come up with a convincing set of data. And what we find is, yes, there is a significant increase, 15 to 20% increase of micronuclei in the cells that lacked the L1, which is the biomarker that we edited using CRISPR. Next slide, please.
Yes, ma'am.
Thank you. So another biomarker of genomic instability, still we are testing by different ways whether or not our protein is linked to replication stress, and slowly the clues came out in terms of 53BP1 foci, micronuclei, and now we progressed to testing lagging chromosomes. So how do we do that? So we culture the cells we plate the cells, and after 24 hours, we culture them in the presence of afidikoline. Afidikoline is a drug that can inhibit DNA polymerase, the replicative DNA polymerase. So it can cause replication fork stalling. So So FD colon after 24 hours, and then after 24 hours of treatment, we fix the cells. So what you see here in green are actually the chromosomes only of the anaphase nuclei. So in your previous slides, you must have seen nucleus as an intact oval or at least an intact round-shaped structure. But in anaphase, you see the chromosomes, right? So you see anaphase nuclei fixed on the slides. So in the pictures, the anaphase nuclei of two different daughter cells. So the wild type and the truncated versions of L1 that we created. Clearly, as seen in the picture, lagging chromosome. So we were wondering, okay, what is a lagging chromosome? I mean, I missed that point. I went straight into describing, bear with me. Pardon me for that. So lagging chromosomes are chromosomes that lag behind at the spindle equator, while all the other chromosomes move towards the spindle poles. So that's what you see where the arrows point. There are pieces of the chromosomes that lag behind while most of it has moved towards the opposite poles. So that's literally because they lag, they lag in terms of their movement towards the spindle poles, you call them as lagging chromosomes. And indeed, quantifications of this data indicated there is a significant proportion of lagging chromosomes accumulation in the cells that carry the truncated version of the protein. So past studies, if you are very interested in studying mitotic defects, you must have come across this already. So studies suggest that pre-mitotic replication stress generates partially replicated chromosomes during mitosis. And this results both in numerical and structural chromosomal alterations. So the absence of the protein that we have been editing caused some sort of pre-mitotic stress in these cells. We're still investigating what is that. Next slide, please.
Yes, ma'am.
Yes, thank you. And I think this is the last test for a replication stress link of L1 that I'm presenting to you all today. Ultrafine bridges. We then went on to test whether or not L1 deficient cells exhibit increased percentage of ultrafine bridges. So ultrafine bridges, to explain you all, are thin DNA threads linking the separating sister chromatids in the anaphase of mitosis. It's again, whatever you see in the panel are anaphase nuclei. So you will see a little red line. We stained a particular protein called PGCHE, P-I-C-H. So this protein actually is found on the ultrafine bridges. That's how we could actually find the presence of ultrafine bridges. It's basically immunostained fixed anaphase nuclei. And again, the cells, if you are thinking about how we did this analysis, we plated the cells, we treated the cells with epidiocolin following 24 hours of treatment. We fixed the cells and then stained them using antibodies against a protein that is specific for ultrafine bridges. So as I said, they are very thin DNA-like threads, not DNA-like, sorry, DNA threads linking the separate sister chromatids. And UFBs, the ultrafine bridges, are thought to form when topological DNA entanglements between two chromatids are not resolved. So again, something is happening in the absence of L1 that leads to some topological changes in the chromatids. Again, this is under investigation. So again, the quantifications here show you the data that is obtained from different clones. So G3C2, G3G7, G3G8. So you may be wondering, okay, is it just in one clone? The answer is no. We have tested in a knockout. We have tested in a truncated version. That's the beauty of CRISPR. The opportunities are endless. As a basic researcher, I mean, I feel there's so much that we can do to answer many puzzles in biology using this technology. So that's a little short story about The first ever molecule that we have investigated, this is ready for publication. Two publications are being written on this at the moment. And we have moved on to other biomarkers as well. I will be very delighted to share with you all at another opportunity. So, oh, there's one more. There's bulky bridges. So, sorry, there's one more. Next slide, please. Is it ready? Okay. Yeah. Is it? When I see someone is trying to talk, I thought, okay, it's ready.
Yes.
Okay. Thank you. So, I mean, just one more thing, bulky bridges. I just missed, I thought I haven't included this. There's another form of anaphase mitotic defect where chromosomes fail to segregate. This is a segregation defect, fail to segregate appropriately and completely during the mitosis. So that's what you see in this slide. Again, the treatment plan is the same, but please pay attention to the representative images here. The wild type cells have segregated and separated very nicely, whereas the delta L1, which is the deletion, the truncated, not deletion, sorry, truncated version of L1, fails to separate. So you can see still huge patches of chromosome being attached towards the late stages of anaphase as well. That is what is indicated in both the images. But there is some amount of bulky bridges on in the wild type. So I can't say that the wild type was completely devoid of the bulky bridges. No, the bulky bridges were present, but the percentage of bulky bridges in the clones that lacked ZFP36L1, the protein of our interest, is more than the wild type. And that's what you see in the quantification that is shown in the slide. Next slide, please.
Yes, ma'am.
Thank you. So what do we understand so far about this biomarker that has been edited by CRISPR in our laboratory? Yeah, with these findings, we are convinced at this point that it is a biomarker of replication stress and a potential drug target. We are currently talking at the moment, collaborating, discussing collaborations, not started collaborating, but discussing collaborations with clinics in Switzerland and in London to test what we have observed in this model in patient samples. So that's going on at the moment. Next slide, please.
Yes, ma'am.
Thank you. So I started this with the talk with the applications of CRISPR, and I want to end this talk as well with the limitless applications of CRISPR. The technology, I mean, I think if anyone starts reading about this technology, will admit that they can't stop getting amazed by the opportunities that this technology provides us to investigate problems, to provide the solutions for the problems that human society faces in the current situations. The best example of how wide the applications of the technology is the CRISPR-based diagnostics for COVID detection. Sherlock Biosciences has developed recently, and it's got approval, emergency use for authorization by the US government to be used in laboratories for SARS-CoV testing. It's a very simple technique. This kit just contains a guide and the Cas9, and this human samples mixed with this kit components leads to the recognition, because the guide is complementary to the genetic material of the virus, and now can bind to the genetic material of SARS-CoV-2, the COVID virus, And upon binding, there's a conformational change in the enzyme which leads to fluorescence, and this fluorescence is emitted is what is detected and leads to diagnosis. So it's amazing. It just takes an hour. So that shows how flexible and wide this technology's applications can be, and it's truly to the imagination and the need of the day. And I hope that you all use at some point or in some way you contribute to the society by associating yourself with this technology. Next slide, please.
Yes, ma'am.
Thank you. So this is a very powerful tool. It has the power not just to edit cells, but also to edit embryos. Therefore, it is a tool that has the power to directly bypass human evolution. Therefore, can be harmful as well to the society. So it's my duty as a scientist working on this extensively to advise you about the ethics of using this technology. There are two schools of thoughts here. People, a group thinks this technology is powerful, robust, use it straight, stop needless human suffering. But the other group thinks that the technology is not ready. We should not be attempting to use this technology to edit human embryos. I believe it very strongly. Unless there are clear guidances, we need to wait for years for such technologies to be applied in routes such as human embryo editing. So there are all, this slide just shows you ban and strict regulations and guidance is provided by different nations for handling this technology in embryo editing. I believe that anything that will cause harm to human society should not be attempted using this technology. That's just a quick advice, because this is another important topic that must be discussed in every CRISPR discussion. So that brings me to the end. Next slide, please. Are we ready?
Just a minute, ma'am.
Okay.
That's all. No?
Yes, ma'am. Change the slide, ma'am.
Okay. Is it changed?
No, ma'am.
This is the last slide, ma'am.
This is the last one, yes?
The CRISPR embryos on the last slide, ma'am. This is that slide actually.
Now there is a slide next to the... Okay, there's no more slide, is it?
Yes, ma'am.
Yes, ma'am.
Okay,
Ethics consideration of...
That's fine. That's just an acknowledgement for the funding agencies, the University of Westminster, Genetic Society, 125 Fund. Without their support, this work wouldn't have been possible. And my entire students, I mean, amazing. I'm so proud. And I'm in this position today because of the amazing, awesome, inspiring, I can use all the best words for my students. They work day in and out, and they are so dedicated in contributing something meaningful to the society. So I'm really proud to be working with my students, PhD students, undergrads, and masters. They all work as a team, and we have recently been nominated, not yet awarded, it came straight away. We've been recently nominated as a team for a Team Excellence Award this year. So we are waiting, fingers crossed. And truly, my students deserve all the honors for bringing this technology to life at the University of Westminster. And thanks to my collaborators at the University of Zurich and Francis Crick Institute, Dr. Raj Radhakrishnan, and to my collaborator, Dr. John Murphy, who is also the Research Director of Life Sciences at the University of Westminster. He has been a huge pillar of support for all the initiatives, holding hands, and a huge amount of support he has provided us in making quick progress. So this was what was summarized with the and also most importantly to the Vice Chancellor, who is also in the picture. I'm not sure why it's not shown. The Vice Chancellor of the university. He's a regular visitor to our lab. He provides a huge amount of inputs and encourages us throughout. So, and also to my wonderful colleagues at the University of Westminster. So I would like to thank you all for your attention, for being so wonderful. This has been, as I said, an emotional event for me. I still have to sit back and think about everything that I heard today. Amazing. It really, thank you again to our Honorable Secretary Vasuki ma'am, and our great teacher, a patient, humble teacher, Dr. Lakshmanasamy sir, and my inspiring colleague and my friend, Dr. Madan, and to you too, Bona, for hosting this event. Thank you.
Ma'am, it's a mind-blowing presentation, ma'am, on CRISPR-Cas technology. I would rather say it is a workshop with detailed methodology and results. Now the session is open for discussion. There are some questions in the chat box, ma'am.
Okay, I'm going to open it.
Yes, ma'am.
Is CRISPR technology a better alternative for GMO foods? I would say yes, purely because of the specificity. We still need to work more on this, but so far the results are promising because it's much more precise in terms of GMO because it is very targeted, very specific. We are not randomly overexpressing the gene of interest in the product. It is very specific and can be controlled. So definitely it is an alternative. Thank you, Ashok. Yeah. Thank you, Deepthi Ashok. Yeah.
Another question from our student Hina Sharif, our third BSc Biotechnology student.
All right.
She wants to know whether we can apply this CRISPR-Cas9 technique to target mitochondrial genome that cause numerous genetic disorders such as neurodegenerative disease, cancer, ROS accumulation, and much more, which has no cure, yet as a result of sensitivity to mutation.
Amazing. What is the name of the student again, Bona, please?
Hina Sharif of third BSc Biotechnology, ma'am.
Hina Sharif, such a wonderful question. Amazing. I'm so really proud to hear such a question from an undergraduate student. Amazing. Efforts are in progress to, as you can see from the presentation, the tool can be targetedly sent to arrive at the nucleus. Still research, there's a huge amount of research that is going on to achieve the delivery in the mitochondrial milieu as well. So I think it's still a very active area of research. We will wait to hear from it more. But absolutely. But because mitochondria, there can be one drawback, Hina. Because mitochondria lacks a very active DNA damage machinery, we have to circumvent that problem. So people have to take more. I think it will involve more procedures to be able to, because as you can see, mitochondrial genome accumulates errors because of the lack of active DNA repair machinery in the mitochondrial milieu. So people have to somehow achieve that. So once that is done, I think we'll be there. So, but there is a lot of research in this area.
She had also asked one more question. I think you have answered already. That what she asked is, what are the challenges for the researchers to make it futuristically possible for this mitochondrial genome targeting?
Yeah, I mean, you need to, first of all, delivery options, delivering the tools. So you have to deliver active enzymes into the mitochondrial milieu. So if you can come up with more efficient methods of delivering both the CRISPR tool and the DNA repair machinery, I think you can move forward from there.
Ma'am, one more question from Karthik Kumar D of our second MSc biotechnology student. Is it possible to use CRISPR technology in mitochondrial DNA disorder? How it works without any bother to conserve genome of mitochondria? Because the mitochondrial regions are connectivity of maternal lineage system. So if it affect, it will collapse the evolution of genes of the species.
Can you repeat the question again, please?
Yes, ma'am. Is it possible to use the CRISPR technology in the mitochondrial DNA disorder? How it will work without any bother to conserve the genome of mitochondria? Because there is a maternal lineage system connectivity, so that it will affect, it will collapse the evolution of genes of the species.
True. I mean, it is. Thank you. Excellent question again. Really excellent question. I'm really very, very excited and proud of you all asking such advanced questions. Really amazing. So, what I would say is, number one, CRISPR is precise compared to many other technologies. So there's no harm. I don't envisage any harm that is caused to the mitochondrial genome. We have to, there's a little bit of off-target effects. I haven't seen much of that in my laboratory because I have designed my own ways of doing things and controlling the cutting efficiency of the tool. So I'm fine with that. But there is also increasing concern from the community that CRISPR can actually create off-target cleavages. So we have to reduce the off-target effects to avoid any unwanted damage to the maternal content. That's number one. And I think now, answering the question of whether or not it is possible, yes, it is. Half of that question has been answered in my previous question, I believe. It is possible, but we need to find out ways of effectively delivering the tools into the mitochondrial milieu. And there's another question here from Arvind Kannan. Thank you, ma'am. Okay. Is CRISPR technology applicable for curing HIV? Yes, true. It is possible to cure HIV by boosting our own immune cells to effectively identify the virus and eliminate the virus infected cells. So there is a lot of research going on in that. Yes, it is possible. Yes.
Ma'am, another question from Manumita. Can CRISPR cure cancer? Is there any limitation in this CRISPR technique?
Curing cancer? Yes,
yes, ma'am.
Of course. Yes, of course. There's a huge area of cellular therapeutics evolving at the moment. So you can actually introduce boosted immune cells again in recognizing cancers. So if you can look at the research on PD-1 molecule more. So yes, it is possible, but it depends on what type of cancer it is. Can CRISPR provide a cure for all types of cancers? The question is still not clear, but definitely for certain cancers that it can provide. Yes, there are molecules that can be targeted. The only biggest disadvantage or the challenge in front of us is how effectively, if you can design effective delivery methods of these tools in the human body, I think you are a winner. Yeah.
Ma'am, another question from our alumnus, Jomi. Can you please discuss about the primers used to generate copies, how to design the primers, and how to isolate the DNA from processed food? Please enlighten us about the CRISPR in world food safety.
Yeah. Again, like any other genetic modification, we have to handle this with care. We should not do anything and everything possible. We should be very responsible individuals. We should act as responsible individuals in using this for modifying food for human consumption. For example, the recent I think addition to CRISPR food is I think it is a salmon. where they have overexpressed the growth hormone receptor, and the salmon has been recently approved for consumption in Canada, if I'm not mistaken. So, but we need to be very careful about for what we use and how we use it. We need more research, and I would focus more on the safety aspects of applying the technology, research enough on the target before I would apply to anything that is allowed for human consumption. There is another question from Sheena. Sorry, Bona, there's a question from Sheena. Oh, sorry, I forgot to, there's one more question that I missed out, I think from Bona, from the student. How do we design these guides? There are a lot of programs. There's a lot of programs freely available at this point. Slowly, it's a big money spinning business at this point. So they're all becoming use and pay kind of a things. But at the moment, there's a lot of free online platforms available. You can make use of them. You can use Synthego, SYNTHEGO, Synthego's platform. That's more. They give more visual representation of guide design. Also, there is another one, Benchling, BENCHLING, Benchling. That's from Feng Zhang, the inventor himself. Inventor of the human genome engineering methods. So there are many online platforms available for designing guides. So you can try at your own ease. Now you guys have time, you can try. Yeah, why not?
Ma'am, one more question from Bersha from Ratnam College.
Yes.
Do we have any disadvantages in CRISPR? Can we use CRISPR technology in fetus? And what are the challenges or errors we face while performing CRISPR?
Yes, fetal genome engineering is still a long way. It's still a long way. It's CRISPR is a relatively new tool. It's just about 6 to 7 years old ever since it was started being applied for various things. So it's a very relatively new tool. So it is a very powerful and robust tool. So we have to be extremely careful. As you can, I think I would like you to read news about the fate of a scientist that used this technology in fetus editing. He has created twin girls using this technology in China, and is jailed for doing that without proper permission. So it's an important question again. Thank you so much. At the moment, the technology is not ready. In UK, we have only one laboratory that has got approval from the Human Embryology Authority to edit human embryos. So what this laboratory does is to obtain embryos from IVF clinics, and then they edit these embryos, and they are not, most importantly, they study only this embryo for 12 days, and they are not implanted in women. So there's a research that is going on in Francis Crick Institute to study the causes of increased IVF failures. So they are only, at the moment, CRISPR is allowed to use only to study the early embryonic defects that lead to IVF failures, and they are not implanted in women. So yeah, we are still a long way from using this technology in embryos. I think we should not start thinking about this at all. At this point, it may be looking very easy. How can I increase my muscle endurance? Can I increase my height? Can I increase my skin tone? Can I do this? Can I do that? But no, the answer is a big no. We don't have enough information about this technology to go ahead and use it for embryo editing. So this is still a long, very long way.
Ma'am, another question from Bhavani. CRISPR technology, is it possible to use to modify the cancer cell to normal cell? Any changes in gene level?
Modify, sorry, again, one more time, please.
Modify the cancer cell to normal cells. Is there any change? Any change in gene levels?
Of course, yes, you can. You can edit. So this is the idea. This is the idea of what I presented today, This is the idea. So can we revert phenotypes? So what the question relates to reverting phenotypes, it is possible to revert phenotypes. Yes. Answer is yes. Yeah. But applying it to the real world, we still have more hurdles to overcome. Yeah. But principally, yes. Yeah.
One more
Go
question
on.
from
Yes.
Shehina Ashraf. When we have a technology like this in our hand, why can't we eradicate cancer or any other dangerous disorders, ma'am? What do you think about this? Is it being difficult thing in this?
Cancer is Sheena, cancer is not just one disease. It is a huge umbrella of diseases, multiple diseases, multiple conditions, multiple genes, multiple mutations. So cancer is like a very big umbrella. So I'm sure we are very close to treating certain cancers pretty soon using CRISPR. But I can't say we will totally eradicate cancer with this technology because of the nature of this disease. So I'm sure, I mean, it is going to be possible. And leukemia, cancers such as leukemia has been treated already very successfully here at the Great Ormond Street Children's Hospital, and in the clinical trials. The clinical trials have shown very, very promising results. Yeah.
Ma'am, one more question from Chellamuthu. Is the CRISPR technology affect the integrity of the original structure of the genome? If it is, it would affect cell longevity.
True, we need to have methods to control the off-target activities of this enzyme. Yes, you're right. If this enzyme goes berserk inside the nucleus, binds in non-intended sites, creates double-strand breaks, it's going to be harmful for the lifespan of the cell. So as long as we can control or limit the cutting efficiency only at the target site, I think we are safe. And I can confidently say that it is possible because I have methods. I have developed methods where I can reduce the off-target effects. As you can see from today's presentation, I have used multiple clones to prove one point. If there was an off-target effect in any of these clones, I wouldn't be able to replicate the same result in all the clones. Yeah, essentially they're different clonal types. Yeah, subtypes.
Question from Meera Darshini. Graft rejection is a challenging part in organ transplantation. Can we use CRISPR technology? How could it overcome such challenges?
Yes, you can humanize the organs. There are also, if you look at the top 20 startups in the US, you will see this is one of the priorities of application of CRISPR technology. Can we use humanized organs? So acquire organs from animals, humanize them, and transplant them. There's a lot of research on that. Yes.
Ma'am, one more question from Karthik Kumar, second MSc Biotechnology. How does CRISPR CPF1 differ from CRISPR Cas9?
Oh, it's the mode of recognition, the mode of the way they cut. PAM is too basic because the Cas proteins are from different sources. The one Cas9 that we use is from Streptococcus pyogenes, just a normal resident microflora of the skin of humans. But it can be from any Cas can be from any source. So it depends on the source. Their properties also vary. Their recognition, their cutting efficiency, their overall structure remains the same. So as I said, the recognition lobe and the nucleus lobe. But then there are minor differences in the way the guide binds and where the PAM, where the cutting location is with respect to the PAM. So all these differ. I mean, very excellent questions, my dears. Amazing. I'm very impressed by the questions that are asked.
Question from Shehina Ashraf. Can we use this CRISPR technology in developing biofuels also? But still,
Yes.
that is also not a much popularized one now. What do you think about the difficulties in this specific point, ma'am?
Again, another extremely important question. A lot of research is going on in this area. For example, microalgae can be used to edit. For example, microalgae are actually not can be are used to produce biofuels. One major disadvantage that was found earlier in microalgae is that there are a set of genes that work against the accumulation of oils in microalgae. So now using CRISPR, scientists have been able to edit the genes that actually regulate this oil accumulation, and they are actually showing some promising results in terms of oil accumulation. Yes, microalgae, editing microalgae is the way forward, and there is a lot of research in that. Excellent question again.
I think there is no more question, ma'am. I would like to highlight some points which you have mentioned. That
sorry, Bhuvana, just quite Meera Darshini. Okay, there's a question. We answered that. Okay, there's an okay. Yeah, we answered most of them. Yes, yeah. So what was that? Sorry. There is a question. Joshua Stephen says cancers are heterogeneous, which is the biggest challenge. That's true. That's very, very true. The more we evolve, humans actually exist today because of their power to adapt, right? We evolve, we find new ways of fighting diseases, and so do the diseases. There's a lot to work, because if you look at the cancer microenvironment, they are very heterogeneous indeed, and leave alone CRISPR, it's a big challenge to any treatment modality. So we still have to overcome. That's an excellent point. Excellent point. We need to still have to keep working on that in this direction. It's a very relevantly new technology. We need more research in these areas. Absolutely excellent point. Heterogeneous cancers are heterogeneous. It's difficult. There's a lot of research in that as well. Yeah.
One more question.
Yes.
What kind of role CRISPR engineering take in conservation aspects in the wildlife society?
Yes, as I said, if we can bring back extinct lives back to extinct species back to life, definitely we can do something about conserving the existing ones as well. So, but I'm not an expert in this area, but I can believe that it is possible. But we need a little more, because these things will narrow down to embryo engineering and stuff like that. So we will have to wait more to be able to apply this in those areas, is what I believe.
I think there is no more question in the chat box, ma'am. I would like to highlight the points which you have mentioned. As you have mentioned, like restriction enzyme, CRISPR also has been invented from bacteria to avoid the virus. RNA protein complex can do wonders in rewriting the genome.
Excellent.
Even we can use this in breast cancer also. And as you mentioned that the breakthrough of specific double-stranded break 3 bases away from PAM motif. And at last you have mentioned that CRISPR also used as a diagnostic tool even in COVID-19 detection, which replaces the need for real-time PCR instrumentation. I'm recalling the days which you explained about the cloning and Western blotting in Indian Institute of Science, ma'am. I'm really impressed. You have proved that you are a best scientist as well as lecturer, ma'am. We need your kind cooperation in setting up this CRISPR technology in our lab.
I would love to.
And on behalf of Kongunadu Arts and Science College, I would like to present a e-certificate as a token of love and gratitude. Ma'am, please.
Thank you.
Accept
Thank
it.
you.
1 minute.
Thank you.
Ma'am,
Oh,
this is
that's,
our certificate.
that's
Kindly.
so lovely. Thank you so much. Thank you so much. That's so beautiful.
Okay, ma'am. Thank you
Oh,
very
that's
much,
so
ma'am.
lovely. I'm so proud. I'm so proud. This is the best part of this. Really, this lockdown period. I mean,
We will send you, ma'am. Send you through email.
that's fine. Thank you. Thank you so much. Thank you.
Okay. Thank you very much. Now I request Dr. M. Gogul Ramnath, Assistant Professor in Biotechnology, one of the organizing secretary, to propose the vote of thanks. Sir, please.
Good evening, all. Gratitude is the most explicit form of courtesy. I humbly take up the privilege in delivering vote of thanks. My heartfelt thanks to our President Dr. M. Archami sir, who has been an inspiration with his dedication for the progress of our institution. I sincerely thank our Secretary and Director Dr. C.A. Vasugi ma'am, a guiding star, who inaugurated this International Alumni Webinar Series and providing her immense support and encouragement for all our endeavors. I would like to express our profound gratitude to our Principal Dr. M. Lakshminaswamy, who facilitates the program and providing his valuable support. I express my heartfelt gratitude to our today's resource person, Dr. Kalpana Surendranath, who spared time from her busy schedule to enlighten us with her excellent presentation on CRISPR technology. I am so delighted in thanking Dr. V. Chinnusamy sir, Chief Executive Officer, and Dr. S. Paulsamy sir, Dean R&D, for their constant support and motivation. I am happy to express my thanks to Dr. S. R. Madan Shankar, Dean Academics and PG Head Department of Biotechnology, and Dr. V. Bhuvneshwari, UG Head Department of Biotechnology, who have been the backbone of this International Alumni Webinar Series. I would like to thank all the faculty members of our department for their constant and moral support. Finally, I thank all the enthusiastic participants for their cordial cooperation. Once again, thank you all. Thank you.
Thank you, ma'am.
Thank you.
I request all the participants to join tomorrow at 10:45 a.m. The next session will be handled by our alumnus, Dr. Gandhi, on Cancer, a tale of incidence and death in India and the world. We request all of you to join using the same Google Meet link. Thank you. Have a nice day. Thank you, ma'am.
Goodbye, everybody. Thank you. Bye. Bye.
Stop recording.